Hi, I'm Paco Nathan from O'Reilly Media, and we're here at JupyterCon, and it's a pleasure to be here talking with Jeremy Freeman. You're now at the Chan Zuckerberg Initiative as the Director of Computational Biology. Mm-hmm. Is that correct?
That is correct.
Awesome. And I'm really curious, I think, to start out with, last time we were talking, you had been leading Freeman Lab at Howard Hughes Medical Genelia Farm.
Yeah.
Can you tell me something about the transition and how this has changed?
Yeah. It's been a big shift. I went from running a lab to, in the span of just a couple of weeks, switching to this totally new role in a philanthropic organization. And on the one hand, it's really different in that rather than sort of being one lab focused on my own research, I'm now in a position where I get to take a much broader view of an entire space to sort of ask how across all of biology can we make all the work that scientists are doing more efficient, more effective, more collaborative. So in that sense, it's really different. At the same time, a lot of the work I was doing in my lab I was sort of a weird lab in that I probably spent half the time on the research I was supposed to be doing, and then the other half was this kind of really broad mix of building and sort of thinking about tools to make the way we do science better. And in some ways, that's all that I get to do now, and that was sort of the thing I was always most excited about. So in those ways, it's been really a perfect and super exciting transition.
Oh, wonderful. Well, how do the tools, what kind of impact do they have on the way that science gets performed? Or, I mean, I guess kind of as background, we were talking before, but people expect that science is this thing that happens step by step, and it's completely reproducible every time, is exactly the same when you do an experiment.
Yep.
And I get the sense it's really not that.
Science is not that, I don't want to sort of make all of science have to be bad. I mean, in science, we are always struggling to make sense of incredibly complex phenomenon, especially in biology. We're constantly developing new technologies to measure new kinds of things, whether it's in genomics or imaging or other areas of biomedical science. So really, science is just super hard and it's super complicated. For that reason, we all really have to work together to both kind of document and organize and share more information about the work that we're doing, and then make it easier for other groups, other labs to reproduce and just understand the work that we've done. So a lot of what we're thinking about is if we take particular areas of biology, so there's this project, the Human Cell Atlas, that we're involved in supporting, can we take one sort of area of in this case, cell biology, and ask and sort of go out and look at what different scientists are doing, how different scientists are making different kinds of measurements, and then figure out through a combination of both funding but also platform development, like data sharing platform development, how do we get the scientists to do and develop their experimental protocols and experimental work in a way that's easier to compare across labs, more systematic, just even going out and taking a set of protocols and running them all in the lab and comparing the results is the kind of thing that in science often we're very driven towards work that will lead to big discoveries and not to do that kind of just systematic, almost like benchmarking of existing methods. And that's a lot of what we're trying to support. At the same time, we have to then take all the data that come from those experiments and analyze it in a systematic and sort of reproducible way.
In terms of protocols, I mean, what is that, like doing lab measurements or?
Oh yeah, it's, for example, there's this really cool new class of techniques called Image-Based transcriptomics, where you are trying to characterize gene expression profiles of individual cells, and you can do it inside of actual cells and tissues by building these special probes that bind to particular RNA molecules with a gene of interest with a little fluorescent tag, and then you capture this image where you see all these tiny spots, and you literally see the locations of where these RNA molecules are. This is super complicated, and it involves chemistry and imaging and optical microscopy, and 10 different labs are developing different ways of doing this. That's super exciting, but it also means there's all these different ways of doing it, and they're all using completely different analysis tools. Every grad student in each lab has written essentially their own custom pipeline using some crazy combination of MATLAB and Python and R and maybe Jupyter, maybe not. So that's awesome. It's this sort of really exciting, frothy world right now, and we want to help accelerate it by helping kind of organize and understand and ideally make a little more consistent and reproducible what different labs are doing.
Wonderful. It sounds like programming back in the day when everybody was running their own assembly language instead of having an operating system.
Exactly.
So now you're building the operating system for science.
Trying to, something like that. I mean, I think one of the challenges is, like, on the one hand, you do want to kind of systematically better organize and understand and sort of make more reproducible and comparable what scientists are doing. But you also want to respect and appreciate the diversity in different approaches. It's good that we have so many people trying so many different things. And I think we're thinking a lot about, and really this requires, I think, working really closely with scientists, doing the work, not to sort of build one platform for everybody, but to instead think about more modular solutions. Take one area, particular kind of experiment where there's a real opportunity to accelerate the way that work is being done, and then work with that community to do that. And that's probably going to look different in different parts of science, for sure. And that's a lot of what we're thinking about right now.
Now for the tooling, for the development of tooling to support this, what kind of cycle do you have? I mean, we talk about doing 2-week sprints or maybe like 90-day releases on a tag for Apache.
Yeah.
But for working with science, like fundamental underpinnings, what's the release cycle like?
Well, for the science itself, I mean, it could be years, right? So there are, in my experience doing actual sort of wet lab experimental work, it's this funny thing where there'll be periods of months, years even, where things are really slow, nothing's really happening, and then suddenly a whole bunch of things click and things start working, and then it's this crazy process where you're like staying up till 5 in the morning analyzing new data because it's so exciting and the field is moving so quickly. In terms of actual software for science, we are trying to adopt kind of best principles of modern software development in terms of timing and cycles. One challenge, but also to me, one of the most exciting things is we are doing a lot of our software development at CZI in collaboration with other groups.
Interesting.
So we're building this data platform for the Human Cell Atlas. That's not, CZI engineers, software engineers are involved and working on it, but we're also partnering with 3 other large institutions and their software engineering teams. And we're all working together almost in sort of an Apache style kind of big open source project. And as you well know, doing these kind of giant open source projects, you can try to have sort of development cycles and sort of keep everybody organized, and we try to do that.
Right, yeah.
But it's this kind of wonderful. Sometimes it's a wonderful chaos.
How is this intended to reshape parts of science? I mean, what's kind of like a near-term outcome for success on this?
Yeah, I think 2 ways to look at it. One is in the short term, there are these areas like the Human Cell Atlas, like these particular areas of cell biology or microscopy, where the near-term output outcome is both data resources, systematic comparison or characterization of every cell in the human body. That will be a resource that people can use to understand basic biology. They can use it to understand as a reference for disease. So these like data resources we might build. I'm super excited about also the software we're developing, whether it's for data sharing platforms or new analyses, that those could be sort of used and reused by other labs for other purposes. I also think a little longer term, something that informs a lot of our work at CZI is that we don't ultimately just want to build tools for any one particular project, but really kind of share and export a process and a way of doing kind of software development for science. And I think that's about doing things in the open and doing things collaboratively. And that's something that if we sort of do it well and do it in collaboration with other groups, that really sets a model for how kind of software and science work together.
It's kind of like the Agile Manifesto for science.
Yeah.
That's maybe another way. I was thinking operating system before, but probably Agile Manifesto.
It's more, it's less sort of one particular platform and more sort of a process and a way of working in sort of computational software problems.
Wonderful. Well, I mean, we're here at JupyterCon. How does Jupyter fit into some of this?
Oh yeah, so many different ways. I mean, there are, to give a kind of a couple examples. Jupyter Hub is something we're super excited about, as many people are, as a way to basically set up an environment for lots of students, postdocs, software engineers to collaboratively work on data analysis. And for a lot of the areas we're working in, there are these really kind of gnarly computational problems. What's the exact right way to analyze or do machine learning on this sequencing data or this image data? And the best way to solve those problems is to get a bunch of great computational scientists in a group and have them work collaboratively on solving those problems. And it turns out Jupyter Hub, as many people have figured out, is a really fantastic way to get 20, 30 students in a room and have them all kind of collaborating in the same environment on the same data set, working together. So that's a super exciting sort of on the collaboration side. in terms of data analysis, I really think Jupyter and the things that will come in the future of Jupyter are a really core part of how we do more interactive analysis and visualization of scientific data.
Any particular areas as far as the future of Jupyter, extensions that you'd like to see, or where?
Yeah, I think a big challenge, and whether it'll be sort of Jupyter or connecting Jupyter with other things, right. is getting towards super interactive, a kind of interactive loop of compute and visualization. Right now, one model is you execute some code and you see your visualization. Right. There are a lot of cases where we want that to be literally as fast as possible. It could be dragging a slider and literally seeing something change in the visualization of the image. Right. Now, things like the widgets or various kinds of web-based sort of add-ons to Jupyter are starting to provide this. This is something my group has worked on, many others have worked on. I sort of feel collectively we haven't quite nailed that yet. Really making it as tight and interactive as possible and fast as possible. And I do think for a lot of more exploratory forms of data analysis, that's going to be really critical.
Sounds like that could go down even to like the API or gateway layer. It doesn't necessarily have to be the notebook artifact. It could be some of the network protocol.
Exactly. And making some of those protocols at a much lower level fast enough to support other kinds of front ends that are not necessarily a notebook, but sort of in the same flavor of the notebook. Right. In terms of having that, it's just super rapid interactivity.
Oh, wonderful. What's kind of the breadth, the scope of the projects that are being funded or labs that you're working with now?
It's broad. I'd say we've existed now for about a year, just under a year, and in that time we have kind of locked into a couple particular areas that we're now working really hard to support. It definitely spans a lot of different parts of biology. There's cell biology, there's genomics, microscopy, imaging, as well as some of these some more general themes and projects around sharing knowledge and science more generally. So things like our support of bioRxiv, the preprint server for biology, is something I'm super excited about as well. So I think just among that, we've sort of bitten off a lot for our first year. The team is very rapidly coming up with new ideas and new areas we're going to explore as well.
I can imagine. I can imagine the list keeps growing.
Yeah, exactly. So in some ways, it's about we really want to. I think as an organization, we want to be known for what we do, not what we say we're going to do. So we're working really hard to frame things where we think we can have a differentiated impact and make progress, and show people that we can really help support some really exciting aspects of science right now.
Wonderful. bioRxiv, that was astounding. I mean, I'm a little bit familiar with archive, but understanding that this is new, biology didn't have this kind of vector for getting papers out.
It took a really long time, and it was really Richard Sever and John Inglis are the founders of bioRxiv. They founded it about 3 years ago at Cold Spring Harbor Lab. It took a while, and it was really their hard work that made it take off in biology. And it took a lot of advocacy and sort of getting people on board. Different communities are different. I think biologists different from physicists, different from computer scientists, different cultures. But now we just said this is something that's working really well, and biologists are super excited about it, so we want to support it. Right. And they were a really small operation and sort of not struggling, but sort of just trying to keep up with the scale as much as bioRxiv was growing became a challenge. So we jumped in and said, we want to support you guys. And that's been a super fun project.
Oh, fantastic. Thank you very much, Jeremy. This is awesome.
Thanks, Paco.
Appreciate it.
So good to see you.
Good to see you, too.